starting with the fluid balance part two, this is more so the electrolyte part of this. And we're going to move into our first two objectives. The main topics we're looking at here are sodium balance and potassium balance. So as I mentioned earlier, we've talked a lot about water so far, how water moves across the different parts of the tubule, how the filtrate is being fine-tuned as that water is being reabsorbed at different points. Now, water is important and we want to conserve water. We talk about the role of that, especially the loop of Henle in that conservation of water. But our electrolytes are important as well, sodium and potassium. And so while we filter these things across the glomerulus, we want to go back and reabsorb some of these things depending on our sodium and potassium state in the plasma. So we'll talk about the various mechanisms for which these two are absorbed. Starting with the first two objectives here, describe how aldosterone and A&P, atrial natriuretic peptide, which are two hormones, how they regulate plasma sodium levels, and then we'll explain the stimuli that releases these hormones. So what triggers the release of these hormones, as well as what so same thing. Objective two is to describe the major mechanisms whereby water and sodium balance can influence the mean arterial pressure. So going back to the blood pressure, the mean arterial pressure that we described in our cardiovascular unit, sodium and water play a huge role in maintaining our MAP. So we'll talk about that connection as well. Now, just to introduce some terms here, as we kind of normally do, hypernatremia is going to be high plasma sodium. That, while it can cause hypertension, is not the same thing as hypertension or hypervolemia, but they're, again, all seen in kind of the same state. Hyponatremia is low plasma sodium, on the other hand. Now, sodium is going to be one of the primary solutes in our extracellular fluid, and its role is important in maintaining our osmotic pressure, as well as the function of our excitable cells. Osmotic pressure in that it creates the gradient that allows water to move. We've talked about this phenomena in several different points so far, but the movement of water across different surfaces and membranes in the kidneys, in the GI tract, that is reliant on the movement of sodium first. And then as far as excitable cells, we know the sodium-potassium pump, we know the electron transport chain, but also our resting membrane potential and our action potentials rely heavily on the movement of sodium. So just to give an overview here of sodium's movement, it is freely filtered, so it moves across the glomerulus with filtration. It's reabsorbed at about 70% of the proximal tubule, distal tubule, and collecting duct. So about 70% of the sodium filtered should be reabsorbed. It's not secreted, which makes sense. That's something that we want to eliminate further from the plasma so it doesn't move into the tubule, rather it moves from the tubule back into the plasma. And then its reabsorption is regulated by two hormones, as I mentioned, aldosterone. We'll talk about how aldosterone is secreted. It's secreted from the medulla, the adrenal medulla, the cortex, adrenal cortex, and A&P, which is released from the heart. So those two hormones will trigger, through the mechanisms that we'll describe, the reabsorption of sodium. And then in the principal cells of the distal tubule and the collecting duct, it is also reabsorbed in a regulated way by those two hormones as well. Now, the sodium-potassium pump is going to be an important part of driving sodium reabsorption, especially getting it across the basolateral membrane. We talked about how it moves into the cell across the apical membrane by either co-transport or anti-transport with other ions. It can move passively into the cell, but then it moves actively across that basolateral surface and then even further into the plasma. So that would be active reabsorption. All right, so just to illustrate that here, in terms of the proximal tubule, remember, it facilitates water reabsorption here. So it moves into the cell freely. This is co-transport. As it moves down its gradient passively, remember it's higher in the extracellular fluid than it is inside the cell. So it moves into the cell passively. And then it can actually help other ions to move against their gradient or other substances to move against their gradient too, right? That co-transport. And across the basolateral surface, it moves via the sodium-potassium pump, where we actively pump three sodium out and two potassium in. So it can move across that basolateral surface and into the plasma via that active transport. In terms of the distal tubule, same mechanisms occurring here, passively into the cell, actively across the basolateral surface via the sodium-potassium pump, and then freely into the plasma. Let's look at some of the effects of aldosterone. So aldosterone, we said, increases that reabsorption, which just means that it helps to increase the number of sodium-potassium pumps and the number of open sodium and potassium pumps on the apical membrane. When we get to potassium, we'll talk about potassium, but aldosterone can influence the movement of potassium as well. Although we're only focusing on sodium here, it actually increases the channels for both. It is made by the adrenal cortex, so the outer, lighter staining region of the adrenal, as opposed to the inner medulla, which is more actively involved in the nervous system, the release of norepinephrine, etc. So the cortex is really where we have the steroids and other hormones being released. And then its action is going to be down here on the principal cells of the distal collecting tubule and the collecting duct. It's going to upregulate the insertion of sodium potassium pumps on this surface, the basolateral surface. It is also going to upregulate the number of open sodium and potassium channels. All right. And in doing so, it helps to stimulate the reabsorption of sodium, and then, as we'll see later on, the secretion of potassium. Now, how do we get that aldosterone, right? What are the triggers to stimulate that aldosterone? And that starts with a long cascade, right? A cascade or a sequence of events beginning with the release of renin from the juxtaglomerular apparatus and the granular cells found in that apparatus. So we've talked about that apparatus before, and some of the cells there secrete an enzyme known as renin. Renin will enter the blood cell, the bloodstream, excuse me, the plasma, and then it will combine with another precursor, which is the angiotensinogen. And the tensinogen, the suffix gen, tells us that it is unactivated. It's in its precursor form. So renin will activate angiotensinogen, which is secreted by the liver, and convert that into its active form, which is angiotensin 1. Now, angiotensin 1 is released or sort of activated in the bloodstream, but ACE is another enzyme that is found on the capillary walls of the endothelial cells. And so ACE actually converts the angiotensin 1 into angiotensin 2, and then angiotensin 2 itself can stimulate the adrenal cortex to release aldosterone. So again, it's a cascade of events that is triggered by things like a drop in mean arterial pressure, triggers the liver to make angiotensinogen, the JG cells of the kidneys to make renin. Angiotensinogen is activated by renin into angiotensin 1. Angiotensin 1 is converted into angiotensin 2 by an enzyme known as ACE, angiotensin converting enzyme. This is found in the capillary endothelial walls, and then that triggers the release of angiotensin 2, which converts aldosterone secretion happening in the adrenal cortex. Now, ACE is especially found in the lungs, and there was a lot of connection if you guys were following the literature and kind of the science around COVID. There was a lot of connection between this enzyme and destruction of this enzyme in COVID, right? The COVID-19 virus. And so that played a particular role in some of the lung responses, tissue injury, tissue damage that we saw as a part of the COVID, right? Because of this presence in the lungs. So just to recap that flow of events here one more time, the liver makes the precursor, angiotensinogen. The kidneys make renin, which activates that angiotensinogen into angiotensin 1. The blood cells, right, the capillary endothelial cells, they activate that angiotensin 1 to angiotensin 2 by releasing ACE, right? ACE is typically released in the bloodstream because of by the endothelial cells. And then the angiotensin 2 is what actually triggers that aldosterone release from the adrenals. All right, let's talk about what the angiotensin 2 triggers. So it triggers the aldosterone release, but it also triggers other things related to a drop in mean arterial pressure. Remember, the original trigger to release angiotensin 2 is a condition like drop in mean arterial pressure, low blood pressure, dehydration, etc. So that stimulates our systemic arterioles to constrict, right? We'll have vasoconstriction in the systemic arterioles, which will directly increase mean arterial pressure as a result. It will also trigger that, again, aldosterone release from the adrenal cortex. That stimulates sodium reabsorption through the mechanisms we described. It upregulates the insertion of sodium-potassium pumps in the basolateral surface and those sodium channels in the apical surface to facilitate the reabsorption of sodium in the distal tubule and collecting duct. And so that increases our extracellular fluid osmolarity. In other words, the sodium content and thereby the fluid content and pressure of our plasma. That will actually trigger the posterior pituitary to release ADH, right? This is another trigger that happens with angiotensin 2. ADH release, remember, ADH is antidiuresis, antidiuretic hormone. And so that increases water reabsorption from those two same places, the principal cells of the late tubules and the collecting ducts, and facilitates that water to be reabsorbed back into the plasma. That is also going to help increase our plasma osmolarity and therefore our mean arterial pressure. And same thing with the hypothalamic neurons. They stimulate thirst and they basically encourage us to drink or intake fluid as a part of correcting that plasma volume and increasing mean arterial pressure. Alrighty. So we've talked about the stimuli for angiotensin 2. What are the stimuli for renin? Renin being released from the granular cells of the JG apparatus in the kidneys. What are some of the kidney responses that will stimulate renin release? So that would be a drop in the pressure in the afferent arterioles. So if the kidneys detect that there's less pressure coming in the blood vessels in the afferent arterioles, that will be a trigger for renin release, which will eventually trigger angiotensin 2 release, which will, through the four mechanisms we just described, increase plasma volume, increase pressure, and correct the mean arterial pressure. So renin is going to be working to correct that mean arterial pressure way downstream. The other thing is the renal sympathetic nerve activity, which is also triggered or elicited in times of um, dehydration or hemorrhaging. And then a decrease in sodium and chloride in the distal tubule can also trigger that as those tubules come very close to the JG apparatus. And then they can sense how much flow, how much sodium, how much chloride is in that distal tubule, and whether or not we need aldosterone in order to reabsorb some of that additional sodium. All right, so here's a slightly different flow chart. This one is describing how a drop in mean arterial pressure triggers renin release, the three mechanisms that we just now described. So a drop in mean arterial pressure Again, we're going to compare this to dehydration, hemorrhaging, extremely low blood pressure, anything under 80 millimeters of mercury. And then the first thing is our after arterial pressure drops, which triggers our JG cells to release renin. Second thing is our baroreceptor reflex. That just triggers an increased sympathetic activity, which further triggers renin release. And then thirdly, the drop in GFR means that there's going to be a drop in sodium and chloride that is coming through in the distal tubules in that filtrate. And so that will, through the paracrine signals that are released by the macular denser cells, that will trigger the JG cells to release renin as well. All right, as that distal tubule comes close to the JG apparatus, it can sense what the content is in that filtrate, and then it can send signals to fine-tune renin release to correct those changes in the distal tubule.